Today's my presentation topic is important classes of virus. Introduction. So next slide. Overview of all viruses and their importance. Animal viruses are viruses that infect animals, including humans. They are diverse and can infect a wide range of animal species, from domesticated animals to wildlife. Animal viruses can cause a variety of disease in animals, ranging from mild illness to severe and life-threatening conditions. Some viruses can also be transmitted to humans, causing zoonotic disease. Importance. Animal viruses are important for understanding the basic principle of virology and for developing antiviral therapies and vaccines. Animal viruses are important for their impact on animal and human health, the economy and scientific research. Studying animal viruses had led to significant advances in our understanding of viral replication, host virus interaction, and human responses. This knowledge has enabled the development of effective antiviral therapies and vaccines against viruses. Classification of animal viruses. Classification of animal viruses on the basis of genetic material, DNA viruses and RNA viruses. Classification of DNA viruses into double-stranded DNA and single-stranded DNA. Double-stranded DNA, first one is herpes virus. These are double-stranded DNA viruses that causes a range of disease in humans and animals, including cold sores, genital herpes, chickenpox, shingles, and mononucleosis. Example: HS virus, VZ virus, and EB virus. Second one is adenoviruses. These are double-stranded DNA viruses that commonly causes respiratory infections, conjunctivitis, and gastrointestinal illness. Third one is papilloma viruses. These are viruses that infect epithelial cells and can cause warts as well as cancers of the cervix, anus, and throat. Fourth one is pox viruses. These are the viruses that cause disease such as smallpox, monkeypox, and cowpox. Next, single-stranded DNA viruses. First one is paraviruses. Paraviruses are viruses that infect animals and humans. The human paraviruses B19 causes erythema infectosum or fifth disease, a common childhood illness that causes a rash and fever. Second one is aniloviruses. Viruses that are commonly found in human blood. They are not known to cause any disease, but they can be used as markers of studying human population genetics. Next, in RNA viruses, first one is negative sense single-stranded RNA viruses. They have a genome that is complementary to the mRNA strand. Example, orthomoxovirus family, which includes influenza A and B. Second one is positive sense single-stranded RNA viruses. They have a genome that can be directly translated into proteins by the host cell without requiring synthesis of a complementary RNA strand. Example, flavivirus family, which includes Zika virus, dengue virus, and yellow fever virus. Third one is double-stranded RNA virus. They have a genome consisting of two RNA strands, which can be segmented or non-segmented. Example, reovirus family, which includes rotavirus, which cause diarrhea in infants and young children. Fourth one is retrovirus, viruses that use a reverse transcriptase enzyme to convert their RNA genome into DNA. Example, HIV, which cause AIDS. So next. Replication of animal virus. First one is attachment. Influenza virus becomes attached to a target epithelial cell. Second one is penetration. The cell engulfs the virus by endocytosis. Third one is uncoating. Viral contents are released. Fourth one is biosynthesis. Viral RNA enters the nucleus where it is replicated by the viral RNA polymerase. Fourth one is assembly. New phage particles are assembled. Sixth one is release. New viral particles are made and released into the extracellular fluid. The cell which is not killed in the process continue to make new viruses. So next, emerging viral disease and their origins, animal origins. COVID-19 virus originated by bats. Ebola virus originated by fruit bats and primates. Zika virus originated by monkeys in Africa. Nipah virus originated by bats or pigs. Hendra virus originated by horses or fruit bats. SARS virus originated from bats. MERS originated from camels. Vaccine development against animal viruses. Rabies vaccine, and mouth disease vaccine, porcine circovirus vaccine, canine distemper vaccine, avian influenza vaccine. These are the structures of animal viruses. Next, next. Next is animal virus control and preventions. Strategies for controlling and preventing animal virus infection. First one is vaccinating. Vaccination is one of the most effective strategies for preventing viral infections. Vaccines can stimulate the immune system to produce antibodies and memory cells that can recognize and neutralize the virus upon infection. Second one is biosecurity measures. Biosecurity measures such as quarantine, disinfection, and proper waste management can prevent the spread of viruses between animals and reduce the risk transmitted to humans. Third one is vector control. Some viruses are transmitted by vectors such as mosquitoes or ticks. Vector control measures such as insecticides and repellents can reduce the risk of transmission. Fourth one is the next. And antiviral drugs. Antiviral drugs can be used to treat viral infections and reduce the severity and duration of symptoms. Fifth one is genetic engineering. Genetic engineering have made it possible to develop new strategies for controlling viral infection. Sixth one is surveillance and monitoring. Surveillance and monitoring of animal population and the environment can help to identify and respond to potential outbreaks. Last one, seventh one is education and public awareness. Educating the public about the risk of viral infection and promoting good hygiene practice such as hand washing can reduce the risk of transmission. Conclusion. In conclusion, we learned that viruses are infectious microbes that are made of protein and genetic materials. Animal viruses can cause infection in animals. Animal viruses infect animals and human causing different viral disease. Thank you.